Hi, how are you? So, um, with this one, I'm going to teach you about the key knowledge about the molecules in our human body. So, what is the main idea here? So, we of course can talk about so many different molecules. Our body has from top to bottom, from outside to inside. There are so many molecules like tears, right? Molecules, fluid molecule in the body, molecule in the cells. So those are important molecules. But with this one, I want to focus on three major molecules. And the reason is that these three major molecules are most important one, most common one, and basically can explain a lot of things in our body. So the three molecules we're going to talk about are carbohydrates, lipid, protein, actually four molecules, and nucleic acid. So four molecules are important. We are going to talk about these four. All right, let me share the screen. Yeah, I think that's this one. All right. All right, so this is it. All right, so organic molecules. So here we are going to talk about carbohydrates, lipids, proteins, nucleic acids. There are one common theme across these four molecules, which is these four large molecules. These are large molecules, organic molecules. These four molecules are polymers. So right here, these are polymers. Polymers means that it's a combination of many monomers. So right here. So a common theme is carbohydrates is a polymer is formed by what monomer? So what are the monomer of it? Lipids is polymers, are polymers. Then what are the monomers? Proteins are polymers. Then what's the monomers to form the protein? Nucleic acids, we will talk about the examples. DNA, RNA are nucleic acids. They are polymers. And what's the monomers? So those are 4 times 2, 8 questions, okay? Guarantee you're gonna see those. So that's that. So you need to know that. And yeah. And another common thing is that even though you have different monomer form different polymer, the structure is very similar. It's all linear structure. So you can kind of think about it's one connect with the other, connect with the other. So it's a string, basically. It's a string, a bit, a string of bits, like a necklace, all right? A string of bits connect together. And so that's a polymer. And depending on what kind of monomer, you have different kind of polymers. So let's start from the first one, carbohydrates. So here, monomer, polymer. So here is just general concept. Here you have one, two, three, four monomer combined with become a polymer. So this is a large molecule. So this could be the carbohydrates, proteins, lipids, etc. And you can break it apart to become monomer. So this is unlinked monomer. So basically this one comes from four monomers. So that's that. And monomer can form polymers. Polymers can break it down to monomer. This is a basic idea about the metabolism. Basically, metabolism means you had the catabolism and anabolism. So anabolism is to structure a larger molecule, molecule. So that is the ana, sorry, anabolism. Catabolism is to break it apart. So that's basically the metabolism are these two. A very typical example is your nutrition. So your chicken is a large piece. You have to break it apart. You have to tear it down, cut it, your beef, cut it, chew on it, grind it in your stomach, break apart. So from the large one to become many small pieces. Then you can absorb them. After your absorption, your body, so that's the catabolism, break apart. In your body, you put them together to form your muscle. So that is the anabolism. You combine them again in the way that you want it. So that is the metabolism. All right. So that is the anabolism, anabolic reaction, and the catabolic reaction right here, anabolic reaction. So that is that. And to break the polymer to break the polymer to monomer, you will need water. So water coming in provide the hydrogen and HO. So H2O provide H and HO, and so that will close up, separate these two pieces. Yeah, so this process is a catabolism, and because it requires water, we call it hydrolysis. So lysis, whenever you see the lysis, you know it means it's a breaking apart. Hemolysis is red blood cell disruption, which is not a good thing. Hemolysis. And yeah, so that's something I can think about. But basically, any lysis, proteolysis is protein break apart. Yeah, so we have a lot of proteolytic enzyme to break apart our protein in our digestive system. Yeah, so hydrolysis, using water to break it apart. And of course, this one require enzyme. Yeah, all of this process require enzymes. If we put them together, we produce water. So in your body, when your body uh, uh, put things together, your body with this process, it also generate water. Yeah, so water is produced, which is a good thing, you know. You don't usually drink water, but our body, when body do the metabolism, it can produce some water. Water can be produced from that. Yeah, so this one is called dehydration because you too, dehydration remove that water to the environment. Synthesis is term for the production, produce, synthesis. Yeah. So this from monomers to a polymer is a dehydration synthesis, is an anabolic reaction. From polymer to monomer, that is hydrolysis breakdown, is a catabolic reaction. Yeah, so terminology, okay, you need to know this. So let's just look at the glucose, carbohydrates. What is the monomer? Monomers are monosaccharides right here. So mono means single. Saccharides is a term for those monomers, monosaccharides. If we put two together, we call it disaccharides. Two is di. If we put more, we can have trisaccharides, but furthermore, we call it polysaccharides, much more, many more. Yeah, so monosaccharides are sugars, basically the monomer of the sugars, and we have the three major form in our body. There are multiple monosaccharides, but the most common ones are glucose. This is a chemical structure. Galactose right here, fructose right here. So these are the monomer. You usually don't get glucose. Actually, you get glucose by chewing on your bread because bread is polymer, and then you break it apart. That monomer is glucose. So you do get glucose by chewing that, biting on that bread, uh, and you get the glucose. Uh, this is better than monomer by itself because it requires a little bit time to break it apart. So your body don't get just glucose right away. It takes time, it takes digestion, which is good because you don't want to mess up with your body's environment. So chew on it, take your time, enjoy your meal. That's a good thing. What people don't really pay attention is when they get fructose. Yeah, because fructose are very commonly seen in our juice. So with that one, you have no digestion. You basically just drink it, and go in to your small intestine very quickly. Fluid digestion is very quick. And it also, because this is already a monomer, it doesn't take any time to be digested. It can be absorbed right away. So by drinking juice water or sugar water or any sweet, basically, it's hardly like I can think about that anything that sweet is, well, maybe diet, like diet, like fake sugar, that probably you can avoid it. Avoid a surge of hyperglycemia, basically increase your blood sugar level. So that is, something you need to pay attention. Don't think that drinking juice is healthy. Don't think that juice is fruit is healthy. Well, eating fruit is good because it takes time to digest. Yeah. But drinking is different, okay? So that's the monomer. So that's the chemical structure. And I don't think people will question you. May might, but I will not, okay? The difference of the chemical structure. All you need to know is that these are the monomer. I might have questioned that. Which of following is a monosaccharide? Glucose, galactose, fructose. Those are the monomer. All right? Yes. So basically, go back to the very original question. What is the monomer of carbohydrates? You need to know that. Yeah. All right. Disaccharides are two monomer combined. So you can have glucose plus fructose. It forms sucrose. That's sugar basically. So sugar is the disaccharide. Um, Glucose plus glucose is maltose. Glucose plus galactose is lactose. So when you chew on it, you basically break it down into disaccharides, and your body need enzyme to break these two further apart. So for people with lactose intolerance are the people who does not have the enzyme to break apart glucose and galactose. And so these cannot be break apart. They will stay in your small intestine, continue move on to the large intestine, continue to move on to your colon, and over there, because you cannot break apart, they are going to become the food for your gut bacteria. And over there, bacteria will use them, and they may cause irritation to your gut, and so that causes the symptom of the lactose intolerance. How do you deal with it? Just eat a little bit at a time. Don't just put a lot of them into the body. Then you probably be okay because we need to feed our guts anyway. We need to feed our guts bacteria anyway, and some of those uh, uh, basically will provide good nutrition back to our body because we have this symbiotic environment between us and the gut bacteria. So that's that. So that's the disaccharides. All right. So you need to know sucrose is glucose plus fructose. You eat sucrose, you produce glucose and fructose, maltose, lactose. Polysaccharides are the polymer of these monomer, and an example would be, so here is the example, glycogen is a polymer of glucose. Where do you see the glycogen? You see the glycogen in your muscle and the liver. When you uh, eat the glucose, catabolism, break apart, absorb them. And then how do your body use it? Your body will not use them right away. So they will put it back. It's an anabolism and then store them. Anabolisms, put them together, form glycogen like this one here. You can see multiple bits. Form them together and then and then store them in the muscle and in the liver. Yeah. So that's the glycogen. So just like this diagram that the process of uh, uh, dehydration synthesis is a process to form the polymer. Hydrolysis is to break it apart. So we can have the similar thing in the carbohydrates. Two glucose form sucrose. Break it apart, hydrolysis form glucose and fructose. So glucose and fructose form sucrose. Yeah. So that's dehydrolysis. Opposite direction is dehydration synthesis. And then this is polysaccharides. This is the and there are several examples right here. Starch, like the bread here. Glycogen is one in our body. Cellulose, those are the dietary fibers that you eat, but you cannot in human body, we don't have the enzyme to break it apart, which is good, you know. We use this cellulose to our large intestine again. So we cannot break apart, just like lactose. If you have lactose intolerance, cellulose will go into the colon, and then bacteria can use them. It also help you to stimulate your bowel movement, basically, allow you to form feces, yeah, which is good for your body. All right, so that's carbohydrates. The next one is lipid. Lipid is also a polymer. And so what's the monomer? The monomers are fatty acids and the cholesterol. Basically, it's fatty acids. Cholesterol is also lipid, but cholesterol don't really form polymer. Cholesterol has different derivatives. So it's a chemical and it can be added on and a little bit change their chemical structure. So that's cholesterol. They don't really form like one become two or three combine together to form a larger one. They don't do that. The reason that we consider cholesterol and fatty acids are lipid is because those are all water, like insoluble in a way. Yeah. And also we can find these two mixed together very well. In our lipids, we always can see some cholesterol, integrated into it. So they don't really form a polymer. Cholesterol don't really form a polymer, but they kind of integrated, like put inside of the situation of this fat. But fatty acids does form polymer. The situation is quite simple. You can have a single fatty acids. Yes, you can have single fatty acids like this one here, single fatty acids. So you can see that you have fatty acids. You have here the head and also multi like carbon chain, and so that's fatty acids. You can have single fatty acids, or if we put them together, you can have two fatty acids, or you can put three together to have three fatty acids. And so for the polymer, we basically has two fatty acids that forms the phospholipids. So phospholipids is formed by how many fatty acids? The answer is two. Triglyceride is formed by how many fatty acids? Answer three. And also it provides you here tri. Okay, so that this one is two fatty acids. This one has three fatty acids combined together, forming the polymer. So that's that. So single fatty acids has a structure like this. So it's very straightforward. Fatty acids has two major form, actually two major structure, two major class. One we call it saturated fatty acids. So this one has very high standard in a way because when you look at that carbon chain, uh, you don't see double bond. You only see single bond, and so that's a saturated. When you put so that's the saturated fatty acids. Unsaturated is that okay? You have double bond somewhere between these carbons. Yeah. So that's the unsaturated. So if I have a picture like this one, ask you that which one is saturated, which one is unsaturated, you can find that if there's double bond in the along this carbon chain, you know that's unsaturated. In our life, uh, these fatty acids would usually not present just by monomer because they usually present as a polymer. So a common polymer is triglycerides. And so triglyceride, let me show you the picture, triglyceride right here. So right here. So that's for here. So this triglyceride, so you have glycerol bind with poly with 1, 2, and 3 fatty acids. So that's triglyceride. Yeah. So that's that. The difference between saturated and unsaturated, what's the difference? All right, chemical structurally saturated do not have double bonds. Unsaturated, you will see double bonds. But other than that, what do you need to know? Uh, what you need to know is that when we take food, we want to choose that in certain situation we want to take saturated fat, in certain situation we want to take more unsaturated fat. Generally speaking, people suggest unsaturated fat more than saturated fat. So it's more preferred according to our current understanding of these fatty acids. The reason is unsaturated fatty acids is not only providing to your body the energy that you can use, fat provide you the energy. Not only that, unsaturated fatty acids also provide the essential fatty acids that your body needs. Essential means that this is the molecule your body cannot generate. You have to take it from the food. So all these essential fatty acids, such as omega-3, omega-6, you see this DHA, you get a vitamin, or you have the diet. You will see those DHA and omega-3, omega-6. Those omega meaning where you see those double bond. So omega-6 is the double bond present in the number 6 carbon. 1, 2, 3, 4, 5, 6. So that's omega-6. Omega-3 is the double bond present in the carbon number 3. So that's the essential fatty acids. And so that's why people prefer to take the unsaturated fatty acids over the saturated fatty acids. All right, now you have this idea that you need to take, you prefer to take unsaturated fatty acids. Then where do you take? And so in general speaking, the saturated fatty acids, because they have the linear structure, this all have linear structure. It's just this one has a very homogeneous single bond throughout the carbon chain. The When you have a lot of these together, they usually packed very well because there's no kink. See, this is a kink. The kink create the space. And so if this unsaturated, it will be more space between, and so it will appear more liquid. Unsaturated fatty acids are usually liquid, right here, liquid at the room temperature. And saturated because this is very straight. So we pack them together naturally. They just pack them together very tightly. So it will be appear quite solid. Yeah. So that's the difference. And liquid usually is vegetable oil or liquid. Animal oil are usually solid. And most of situation, in most cases, that's that. So if you people want to take the unsaturated, they prefer, to take it from the vegetable oil. Yeah. Essential fatty acids. Remember that we're gonna have quiz on this one. That is the unsaturated. All right, so that's the monomer. Another form would be called for the double bond. We can have a, cis cis location or cis formation of two hydrogen are located in the same site. This is called cis. We can have cis configuration. We can have trans means that two hydrogen are in the opposite direction, and so this is trans. So we can have a cis unsaturated fat or trans unsaturated fat. And this one is natural. Trans is through some cooking process or manufacture process, you can convert this one to trans. Yeah. So that is that. So this is not good. Trans fat is bad because this is not natural. Cis fat is better. Yeah. So that's the monomers. Fatty acids are monomers. They can form polymer through dehydration synthesis, a anabolic process. So when you have two bind together, they will bind on glycerol. So this is a glycerol, two bind on it. Glycerol provide three binding sites, two bind on it. Then what is used to bind on the other one? Uh, Several things. A common thing is just a phosphate group, like this one here. And so this is called phospholipid. And phospholipids, you have two fatty acids and one, yeah, so it's polymer of two fatty acids. Where do you find the phospholipids in our body? You find this very common in our cell membrane or plasma membrane. Mm. So, so with this one, you have two tail. These are fatty acids, so this is lipid. One part is not lipid, and this one has some charge on it. So this is more water friendly. These are water phobic. So this is hydrophobic tail, hydrophilic head, hydrophobic tail, two tails. And so these can be used and commonly seen in our cell membrane. What else? So this is also called diglyceride because it has two. This glycerol compared to triglyceride, right? Triglyceride, we have a three. So here is the membrane. This is your cell. In our next lecture, we're gonna talk about the cells. And so the cell membrane is this. You have one layer of the phospholipids, hydrophilic, like high water, hydrophobic against water. So this hydrophobic will very happy to meet another hydrophobic tail. So these two tails will naturally uh group together, just like lipid. If you drop them in the water, they basically, they will combine. And so this lipid will group together, but because there is a hydrophilic area, so they will align together. And so with this phospholipid on one side, another phospholipid on the other side, you can have hydrophilic on one end, hydrophilic on the other end. And with this one, this is hydrophobic, this is lipid layer. And so you basically separate your water into two compartments. And you can have continuous on this one, then you can form a cell membrane. So water outside, water inside. Outside water, we call it extracellular water. Water inside, we call it intracellular water. And it's separated by this phospholipid. This one is also sometimes it's also called uh, bilipid layer. I'm not so sure if it shows here because it has two lipid, two lipid layer basically, lipid layer here, bilayer, phospholipid bilayer. Yeah, so that's that. All right, what else? Hydrophobic molecule pass through freely right here because this is lipid, so lipids can go through it easily. Lipid soluble stuff can go through that easily. However, because this lipid, your water soluble stuff, anything that you can mix in the water will not pass through because here is against those water soluble stuff. So they can separate the water uh, into compartments or water soluble stuff into compartments. So that's that. That's two fatty acids. What if you have three? When you have three, this glycerol can let you bind three. So that's the maxima you can bind on it to it. And the three we will form this triglyceride. So you have one, two, three fatty acids. You can have saturate, you can have unsaturated, and yeah, so fatty acids combined with the glycerol, this process will become dehydration synthesis. You can then break it apart. Our enzyme can break them apart, so we can use fatty acids when we need it in our body, or when you digest, when you eat fat right here, when you take some like a vegetable oil, it's triglyceride. Your enzyme will need to cut it and to become fatty acids, and then this can go into your body. So that's that. This is how our body do it. So when you eat it, you break it apart. In your body also put them together and then store them in your fat tissue or adipocytes, adipose tissue. Yeah. So that's the fat. Let's take a break and then we will talk about the other two molecules.